So welcome to the seminar or to this colloquium, and in particular, welcome Professor Jochen Guck as our speaker today. So Jochen Guck is director of the Max Planck Institute for the Science of Light at the Max Planck Center for Physics and Medicine in Erlangen. And he's also professor for biological optomechanics at the Friedrich Alexander University in Erlangen. So before I give the screen to Professor Guck, let me briefly introduce him. So Professor Guck is from education is a physicist. He studied in Würzburg, and he received his PhD in physics from the University of Texas at Austin in 2001. And after that, he was a group leader at the University of Leipzig and moved then 2007 to the Cavendish Laboratory at Cambridge University as a lecturer and a reader later on. And in 2012, he became a professor for cellular machines at the Biotechnology Center of the Technical University in Dresden as an Alexander von Humboldt professor. And since 2018, he's at the Max Planck Institute in Erlangen, which I mentioned. So his research interest are physical properties of biological cells and tissues, and in particular their influence on cell function and behavior. He's developing for his studies, he's developing novel photonic and microfluidic and other techniques, and he's also interested for applications of these findings in diagnostics, medical diagnostics, and therapeutic approaches. So he received several awards and prizes for his work. I will not mention this in detail here. We are very happy to have you here, or to have you online, at least to give a presentation in our colloquium. And I'm keen to hear you talk now. Professor Guck, the floor is yours.
Well, thank you very much for this introduction and also for the invitation in the first place. I didn't know that the PTB has an Abteilung that is interested in this kind of research. So until a few months ago, until you contacted me, and it's great to see that. And I was really looking forward to visit you in Berlin to also see what you're doing, but at least I can show you what we are doing now. So as you said, I'm a physicist interested in physical properties of biological cells. I thought I'd start with a research overview, but you've also kind of said that already. So most of what we're doing is centered on mechanical properties of cells, what we can learn from a mechanical measurement. We're also interested in how cells themselves measure and respond to the mechanics of their environment, in particular in the central nervous system. So to what extent, I don't know, a scarring after an injury in the CNS, for example, prevents regrowth of axons because the axons might sense that there are some stiffness changes ahead of them. That's also how I know Ingolf Sack, because obviously MRE is one of the techniques to study that in situ in humans, mechanical changes in the CNS. We've also had quite a bit of fun in measuring optical properties of cells, so not just mechanics, also optics. That's in particular relevant in how light propagates through the retina to the photoreceptors. We had quite some fun, as I said, in figuring out that there are so-called Müller cells that act like optical fibers, and the nuclei of the photoreceptor cells have a very peculiar chromatin arrangement to facilitate light transport through that neuronal layer to the photoreceptors, which are buried underneath all these neurons, actually. And we also develop a lot of the tools that we need to do our research ourselves. So today I will be mostly talking about, or exclusively talking about, cell mechanics and tool development, and not touch upon the other two here. So the basic question when it comes to cell mechanics that drives our research is essentially, what is a cell and what is its function? How can we tell from the outside what a particular cell is and what it's doing. And of course, in the day and age of molecular biology, with all the genomics, transcriptomics, proteomics approaches, you can get essentially the complete molecular information about a cell. And within that information, clearly, is also the key to understanding what a cell is and what it's doing. Right, but there's the obvious problem that this is a very complex situation. Several 10,000 different kinds of proteins that could be relevant, and there are different expression levels and interactions, and it's a very complex system. So I think we're still far away from inferring from this molecular level information what a cell is. Even though it's there, it's hard to interpret, and practical problem is once you have that information, the cell is not there anymore because you need to take it apart. And so to get around this more practical problem, people have figured out that there are certain proteins that are existing at the surface, receptors and other proteins, protein clusters that you find at the surface of cells so that you can attack them from the outside without taking the cell apart. You do that with fluorescent or magnetically labeled antibodies. And these proteins are then characteristic for a certain cell type or a certain function. A couple of potential problems, don't want to talk this down. I think this is a super useful approach and obviously very widely used. But a potential problem could be that by binding something to a protein that's at the surface, you're also talking to the cell, right? Proteins have functions, they are there for a reason. And if you're not careful and you don't know what particular part of the cell to engage with, what epitope to use, for example, you could actually be signaling to the cell. That means you're changing the system. That's a minor problem. You can get around that. There's certainly a practical aspect of requiring a certain amount of time to prepare, and in particular to have certain costs. Antibodies are expensive. So you need to do something from the outside in order to extract that information. And then a more fundamental problem, I think, is that, and a more restricting problem is that you need to know already what you're looking for. Right? You need to know which protein you need to label in order to tell whether that cell is, I don't know, a hepatocyte or a leukocyte or whatever. So what if you are faced with an unknown situation, with a cell that you don't even know, where you have not established that link between a set of proteins at the surface and what the cell is doing, or its function? What if the cell starts to change its function but still maintains the same kind of protein at the surface? You don't know that. You're blind to that. So you need a lot of prior information in order to make that work well. And that's kind of where our approach comes in, because we say, what about forgetting all of this molecular detail? What if we say the cell is a physical object? It has certain properties, certain physical properties, like mechanical properties, which ultimately integrate all of the molecular information into an easily accessible physical property. Right? All of this will translate into the cell having an elastic modulus or a certain viscosity. That's interesting for a number of reasons. One is that this is completely marker-free. The cell has the properties that it has. You don't need to do anything. You just need to measure that. It's also not as hopeless as that might sound in the beginning, because in particular, the mechanical properties are linked to cell function very intimately through the cytoskeleton. The cytoskeleton is a polymer network, cross-linked, very dynamic, actively regulated by the cell, that to a large extent also controls the mechanical properties of the cell. And the cytoskeleton is also very heavily used by the cell in all kinds of different functions, right? When it wants to migrate, when it wants to divide, when it wants to transport, endocytose, whatever, it needs to rebuild the cytoskeleton in some way. And that means the mechanical properties change. And it doesn't matter what the cell is doing, as long as it's changing the cytoskeleton, you will be able to feel that from the outside. So that's a very sensitive approach. And this is also something that addresses many of the points that are brought up at the top. It's non-destructive, you don't signal to the cell, you don't have any extra cost because of labeling. You don't even need to know what you're looking for. You can just measure and see whether anything changes. And this is not too dissimilar from something that you're doing from your everyday experience, in that if you go to the supermarket, and Yusuf will probably be really bored to hear that yet again, If you go to the supermarket and buy tomatoes, of course, you use your visual senses to find them. They're red and round and whatever. But then what you do is you grab that thing, you squeeze it, and you check whether it has the right stiffness, right? Whether it's too soft and mushy, then you probably will let that go. Or if it is ripe and it has particular characteristic stiffness. So this is something that you do anyway. You're all familiar with that. And we're kind of trying to take that approach down to the cellular level. Of course, that requires certain tools, certain mechanism, techniques with which you can measure that reliably. Cells are small, you need to be precise, you have to have a certain amount of force, etc. And so one tool that I've had the pleasure of co-developing and discovering in Austin, Texas already during my PhD thesis, which I did with Joseph, is this microfluidic optical stretcher. The idea is relatively simple. You take two laser beams, point them at each other. That's an optical trap, so you can trap small things in the center. We found out when you send a lot of light in, the cell is actually stretched out, like in this cartoon. That's why we call this an optical stretcher. You record the deformation of the cell with video microscopy. And on the right here, you see a movie running now where such a cell, that's I think this was a fibroblast at the time, you see that it's being deformed, elongates. And that's the response to stepwise increase in stress. And then the stress is kept constant just by turning up the power to a certain level and then reducing it again. And then the cell displays a time-dependent response. It elongates, and that relative radial elongation along the axis, along the horizontal axis, is then recorded. That's a typical creep compliance response of a viscoelastic material, which we can then analyze. So you can do nice viscoelastic measurements at the single cell level. And it's also the fact that this is an optical trap also helps in delivering cells to that measurement, because they can be in suspension. You just flow them in gently one by one, and then they're being trapped and centered, and then you do the measurement and you can extract your information. So it is a really cool tool, and we've been using it 20 years now to do all kinds of measurements and all kinds of cell systems, functional changes. I just plot a couple of them here. When a cell goes through the cell cycle, it changes the cytoskeleton and it has different viscoelastic properties. When blood cells differentiate, they will acquire very different and telltale viscoelastic signatures that will even let you tell apart a neutrophil from a monocyte, for example. Or if you look at malignant transformation, if a healthy cell becomes cancerous, it tends to become softer, quite a bit softer. And the more aggressive the cancer is, the softer the cells are. So, that's something that you can pick up because the cytoskeleton changes. Or immune cells, when they become activated, they very rapidly change their mechanical properties, also because they need that in order to extravasate and go into tissues and fight disease, etc. So the take-home message from that slide is that essentially any physiological or pathological process that involves the cytoskeleton can be monitored by a mechanical measurement in the sense that I introduced a few slides earlier. So you can feel for functional changes in cells. Great. If that works so well, why isn't that widely used? Why isn't that a standard approach similar to FACS, for example, to characterize cells? There are many applications, but potential applications in regenerative medicine, cancer diagnosis, infection monitoring, a couple of others. Why is that not done? And I think the answer to that lies in the techniques that have been used up until a few years ago to do these kind of measurements. So I introduced this optical stretcher already. I think the gold standard at this point is atomic force microscopy, where you have a cantilever and some usually colloidal probe underneath, and you push into a cell and you see how far the cantilever deflects, so you know the force and you see how deep it goes in. And with that, you can again extract viscoelastic properties of cells. An older and still widely used technique, is very robust, is micropipette aspiration. So you have a small pipette, you put that on the surface, you apply negative pressure and suck parts of the cell in. Again, you know the pressure, and then you can measure the response of the cell, elongation, and you extract viscoelastic properties. These are all really awesome techniques. and very robust and very useful, but they are really slow. If you can measure 10, 100 cells an hour, I think that is already pushing it, right? Which is good for basic science, but it's not really good for translation in the real world application. And I really like this statement by Sid Brenner, said, developments in science depend on new techniques, new discoveries, and new ideas, probably in that order. So we need the appropriate techniques, new techniques, in order to open up new areas of discovery and new ideas. And that's exactly what we have done in recent years. So we have taken on this challenge, this question, and have come up with a solution that addresses that point. This is one of several techniques that have been proposed and introduced in the last couple of years that all rely on microfluidics. And we actually have recently published a comparison paper with colleagues at UCLA, Dino Di Carlo and Scott Manelis at MIT, who have different variants of microfluidic approaches to measure cell mechanics in high throughput. And they all perform well. They have particular strengths and weaknesses. Some are faster, some are slower. And the measurement rate actually has an impact on what you can measure. There's such a thing as measuring too fast. If you deform a cell really, really fast, then the actin cytoskeleton fluidizes because of the measurement. And then you're not sensitive to changes in the cytoskeleton anymore. You can still measure chain differences that are telltale, but they might be due to nuclear mechanics or other properties of cells. Anyway, so there are many techniques out there. Ours is called real-time deformability cytometry. So cytometry is clear. We want to measure cells. And in particular, we want to measure the deformability of cells. And real-time will become clear in a second. So the idea is, rather than looking at a stationary cell that you either have on a surface or on a micropipette or in this optical stretcher, and then you do a measurement, you use the motion of the cell to exert stresses. So the cell has to move in order to deform. And in this particular variant, we use a syringe pump to push a suspension of cells through a microfluidic chip that has a narrow channel. It's about 20 by 20 microns, slightly larger than the cells. They're not in contact with the walls as they're being pushed through, but they move with about 10 centimeters per second through a 20 by 20 micron size channel. So you can imagine there are considerable hydrodynamic stresses that are acting on that cell. And then you just need a fast camera to record that motion. And when you do that, this is how that looks like. You see an initial spherical cell that moves into the channel, elongates, and then eventually reaches this steady state deformation towards the end of that channel, which looks like a little bullet or something like that. Right, so I think this is really a mesmerizing movie. You can really see how the structures inside of the cell even deform. And that's really, really cool. But we call this, so what we do is we take images, movies or images, and then we detect whether there's a cell present in a particular frame, and also determine the contour. You see the red line here, that's the contour of the cell. And then you can calculate things like the cross-sectional area, so how large is the cell, or you can quantify the deformation away from a circle, which is a measure of the deformation in the first instance. The deformation, kind of a measure of strain. And because we know we have analytical numerical models that we use to calculate the stress around cells, acting on cells in this particular configuration, given the viscosity of the medium, the confinement, the size of the object, the shape of the object, etc. We can then again use the stress that we know and the deformation that we measure to extract something like a Young's modulus, for example. And it's called a real-time deformability cytometry because we can analyze these images in real time. So a few thousand frames coming from the camera to the computer, normal CPU, 6-8 core, sufficient to process the information and monitor and analyze the cells in real time. So that means now if I show you a screenshot of the control software of the GUI, this is a plot of where every single cell, every single dot that appears here is a cell that has been measured. And what is plotted is the size versus the deformation. So you see there's a distribution of these two properties in a given population, but that characterizes the mechanical fingerprint of that sample. And this is run as fast as we measure. There's a cell counter here, so within a few seconds, we measure a few thousand cells per second. So this is a few hundred or up to 1,000 cells per second. This is a continuous measurement. We just let this run. This can run for hours, and you acquire millions and millions of data for the cell population if you want to do that. That's just so much faster than anything we've done with, or you can do with an optical stretcher, AFM or whatever, that we've completely stopped using any of the other techniques. I mean, this is, we can measure in a single day as many cells as with any of the other techniques in 100 years, right? This is several 10,000 times faster. And so it has opened up an entire new range of experiments that you can do, things that you can test. It doesn't take any time to measure. This has been a lot of fun in the last couple of years. We published many papers. I want to focus on one particular application where I think that also brings out nicely why this is so cool. And that is the analysis of blood. So if you take a drop of blood, really just, I don't know, a few microliters, tens of microliters of blood, you dilute that in some measurement buffer, and then you send that through the chip. This is what's shown here. The cell suspension comes in from the left. So it's kind of a zoom out of what I showed you earlier. There's some clear sheath liquid that is also pushed in here to help center the cells into this channel. And that's the narrow channel. And at the end of this channel, we then measure. And we need to have this kind of throughput for few hundred cells per second, because only 1 in 1,000 cells is something else than a red blood cell. Red blood cells are so abundant that you probably only see red blood cells in this image. And every now and then you also get a white blood cell, and that's the interesting cells. It's the immune cells that you eventually want to get at, right? If you want to look at, say, patient status or something like that. I let this run just for you to get a feeling for how fast that really is, what it means to measure 100 cells a second. So this is really the tool that you need to measure whole blood. And what do you get? This is a characteristic, a very typical distribution of the cell properties in blood, or what you could call a mechanical fingerprint of blood. So there are a lot of cells up here, and then there are a few other cell types, groups of cells down there. What are they? The cells on top, they are relatively easy to identify. Number one, there are so many that can only be red blood cells, right? And also from the shape, they're very strongly deformed. Cells have a disc-like, biconcave disc-like shape anyway in resting state, and are pulled out into a teardrop shape in this channel. So very strong deformation, very high up in this plot, which also means it's very nice that it kind of lifts the wheel to expose other cell types. And the small cells here are the platelets, thrombocytes. And then there are two other populations. The red ones here are the myeloid cells. So everything that comes down the myeloid differentiation lineage. That's the cells of the innate immune system, the first responders when there's an infection or something going on. These are the cells that just go in and try to attack the pathogen, call in other cells for help, etc. And then these are the lymphoid cells, the cells that are coming down lymphoid differentiation lineage, T cells, B cells, etc. And these are the cells of the adaptive immune cells system. So the cells that learn how to deal with a particular pathogen, so that upon second infection, they then know what to do and can be much more efficient and much faster. So these are also the cells that, of course, are playing an important role these days in terms of vaccinations and immunization against the SARS-CoV-2 virus. Okay, 1, 2, 3, 4 different cell types. Myeloid cells consist of neutrophils for the most part, but also monocytes, eosinophils, and basophils. But now there seems to be one cloud. That's slightly disappointing, but we're only looking at 2 dimensions so far, right? Size and deformation. What if we take into account one other parameter that is easily accessible, and that is how bright the cells appear, or conversely, how strong their absorption of blue light is. And erythrocytes are full of hemoglobin, so they absorb the blue light that we use for illumination. That means they appear dark, and other cells are also relatively dark, and others are much less so. So if you now start plotting instead of cell size and deformation, again, these are the 4 cell groups that we can identify: red blood cells, platelets, lymphocytes, and these are all the myeloid cells. If you're now plotting cell size versus brightness, you see that this cloud of cells nicely stacks up to identify or to reveal the 4 distinct cell types that make up the myeloid cells. So these are the neutrophils, these are the monocytes, these are the basophils, and these are the eosinophils. Okay, so we have now 3 parameters that we easily get out of an image that give us enough distinction capability to identify 7 different cell types. And these are also the things that normally a doctor would want to know. If you give blood or somebody takes blood, you're sick, doctor wants to know what's going on. They count how many of the different cell types there are. That's called a blood count, complete blood count. And then if you compare what we are seeing in blood and what a conventional blood cell counter like a Sysmex machine would give you, you see that the values match up very nicely. So we can very easily quantify how many of the different cell types are in blood. But in addition, we also know for every single cell measured for all the different cell types, whether the cell has the right size, whether it's too large or too small, and whether it has the right deformation, whether it is too soft or too stiff, right? And that's for each of the 7 different cell types. And that, you can imagine, spans up a relatively large dimensional space into which we can now try to map different kinds of diseases. So not just how many cells there are, but also whether they have the right properties. So this is going already very far into an application, potential medical application of the idea that you can feel for functional changes in cells. I want to very briefly show you the results of a study that was a lot more extensive, where we went through the different cell types to show that in different diseases, different cell types respond. It is well known that in a malaria infection, as a part of the life cycle of the Plasmodium falciparum, in this case parasite, that they spend inside of red blood cells, and people have already shown, using some of the techniques that I mentioned earlier, that that leads to a stiffening of the red blood cell. So we should be able to see some mechanical change in Plasmodium infected blood. And that's kind of what you see here. This is again a distribution of the red blood cells measured in healthy control. And this is the same measurement in the blood of that is infected with Plasmodium falciparum. And you very clearly see there's a shift of this, this is the main circular contour kind of encircles 90% of the cells. In a healthy control, this would be up here, and there's a large fraction of cells that are actually too stiff. They're not deformed enough, kind of what we were expecting. But now you can also you look at the image, you see in some of the images this white spot here. I hope you can see that. And that's the vacuole inside the cell if there's a parasite. So we can even see and count the cells that have a parasite in them. And these are marked with a red dot here. Maybe you can see that too. So they're kind of all over the place a little bit and not very many. So not that there are cells, there's a mechanical change to the red blood cells, even if there's not parasite in that particular cell. So we can pick up the infection with Plasmodium, number one, by identifying the cells that are infected, but also by seeing a population effect in the mechanical properties of these cells. And if you now compare a throughput of a few hundred cells per second, where you can just look at and acquire this kind of information, to a standard diagnostic approach where you take a blood smear and you put that on a microscope and then you look for the parasites in the red blood cells, that gives you an idea of where this approach could be useful to automate and much faster and probably more reliably identify whether a patient has Plasmodium infected red blood cells. That's the red blood cells. We also looked at the white blood cells. And that's the point where you need this really high throughput. Again, a small glimpse into what we've done in this paper here, published in eLife years ago. What is shown here in black in the back is the distribution of the neutrophils of a healthy donor. Again, plotting cell size versus deformation. And in addition to that, we're also plotting kind of on top of it, the neutrophils measured in a patient with an acute lung injury. And you see there's a characteristic shift in the distribution to larger cells and more deformed cells. And that's in one patient, one case where we had a few hundred cells measured. If you take the median of that distribution and you plot that for 10 healthy donors, this will be the 20 healthy donors. These are the 20 gray dots here. So that's where neutrophils typically are in their mechanical properties. And then you see also 10 or so patient blood samples measured, neutrophil medians for respiratory tract infection and acute lung injury. And you see a very clear shift, not just on this single example on the left, but in general, to larger size and more deformation. So this very clearly seems to be a characteristic signature of an infection going on in the body. And that is, I mean, these are bacterial infections ultimately, maybe triggered by viral infections, but then taken over by bacterial infections. A pure viral infection would look different. The neutrophils wouldn't respond very much, but the lymphocytes would start responding, which we don't see in these respiratory tract infections in acute lung injury, for example. So that gives you also maybe an idea that this could be used to distinguish different kinds of infections, different origins from maybe a bacteria from a viral infection, which could have a really interesting impact in the light of the growing antibiotic resistance of bacteria. Right? If you specify, right? Okay. Sorry for that. To only use antibiotics in the case where it's actually confirmed bacterial infections and not when it's caused by viruses or fungi or something else. What is also easy to do is to detect new cells in blood. For example, in the case of leukemia. Leukemia being characterized by the appearance of immature blood cells that are released too early into from the bone marrow into the bloodstream. So you suddenly get a huge population of cells that would not be anywhere close to what we expect of any of the other populations to be. This is where the lymphocytes should be normally, and there aren't that many, but there's a huge population just outside of that target area. So this would be one way to diagnose leukemia, but of course there are many different ways of doing that already very reliably. But what you can do with this technique very easily is then monitor the treatment success. Right? Over the 12 days of hydrocortisone treatment, you take blood every day and you measure and you see how the population, the properties of that population start to normalize and eventually end up exactly where the lymphocytes should be. So you can monitor treatment success. Right? This is a few examples of what we've done to show that you can diagnose disease, different kinds of human disease conditions, and you can also monitor treatment success. A very wide, potentially very wide range of applications of that approach. And we were in the process of scaling that up to screen tens of thousands of patient samples in the hospital here to then use machine learning approaches to correlate our measurements with the patient outcome, etc., to identify areas where this could be particularly useful to use this kind of diagnostic approach. When then this SARS-CoV-2 pandemic happened, and we got our hands on COVID-19 patient samples, so patients that were suffering from acute COVID-19. and started measuring their blood. And what we found was really striking. So now again, going through various cell types, red blood cells. Again, healthy controls, that's the distribution we would expect. That is the situation in a patient with an acute COVID-19 infection. And I think you can see very easily that this distribution doesn't look anything like it should be. In particular, there are also a lot of cells that are not very much deformed, that almost stay round and don't have this characteristic teardrop shape that you would expect them to have in a healthy patient. After the acute phase resides and the patient recovers, the mechanical properties eventually also return to normal. So that's red blood cells, very different than they should be. Neutrophils. Again, this is the distribution that we would expect them to have. That's the distribution of the neutrophil properties in COVID-19. Very clearly much larger than they should be and more deformed in this case. But again, from what I told you previously, to us, that means there's very clearly an infection going on. I mean, no kidding, right? These were COVID-19 patients. There was clearly an infection going on. Afterwards, most of the patients recovered and the blood cells normalized again. Monocytes typically have a certain volume. In the acute phase of a COVID-19 infection, they're much larger. Right? They're 20% larger than they should be. And then that they go back to normal. Or lymphocytes typically have a particular Young's modulus. In the acute phase of that infection, they are much softer than they should be. Eventually, that goes back to normal. So you clearly see that in contrast to what I showed you before, where maybe one or the other cell type showed some signature, here all of the blood cells have very abnormal mechanical or physical properties. So it's very clearly something severe going on in these patients. But in addition to that, these physical changes, functional changes, might actually be involved in how the disease progresses, because your cells, blood cells that are circulating through your body, if they are too large or too stiff, they probably don't pass as quickly and as easily through the microvasculature of the lungs. So maybe the blood cells that are not deforming enough cannot pick up enough oxygen, and that's some of the fatigue problems that these patients have. Or the neutrophils, they are too large and they get stuck and start to cause secondary problems in organs where they then cause infection, local inflammation, etc. So maybe the physical properties are, in addition to everything else that's going on, involved in how that disease actually progresses. And to show that and to illustrate that better, the connection between what we measure and circulation, I briefly switched to a different cell type. Mesenchymal stromal cells are used for regenerative purposes or immune modulatory purposes, where they're injected systemically and then supposed to grow organs and things like that. And there are different ways of growing them. You can either grow them typically on a 2D culture plastic dish, or you can grow them as a 3D culture hanging drop, where they are in contact with each other, not with a hard substrate. And when you measure the two different ways of growing cells, you find that the ones grown on 2D plastic are large and not very deformed, i.e. stiff, and the cells grown in 3D are relatively small and much softer. That probably has very drastic implications on how these cells circulate. And that's something that we've tested in vivo and in vitro with a so-called microcirculation pneumatic, so a microfluidic channel with successive constrictions. Approximately the dimensions, 5 microns constrictions, and the number about 200 or so that the cell encounters when they pass through a human lung. And when you send these two different cell types through, you very clearly see a difference just by looking at these movies, right? The stiff and large cell takes its time, whereas the soft and small cell is much faster in circulating through. I mean, this is not too surprising. That's what you would expect, right? Small and soft cells circulate better than large and stiff cells. But that's in vitro. Does that also work in vivo? And for that, we resorted to a mouse model where we injected human mesenchymal stromal cells into the tailvein of the mouse and then recovered different kinds of organ, looked at the amount of DNA, human DNA content. And whereas in a typical situation where the MSCs are grown on a 2D culture plastic, you inject them into the tailvein here. On first passage, the first thing that the cells do is get into the lung and then are filtered out by the lung. 80% of the cells get stuck there. This is what you see here. The stiff and large cells mostly get stuck in the lung and then don't make it to any of the other potential target organs. But if you use soft and small cells, a lot less of them get stuck in the lung and they get a much better chance of actually reaching the target organs. So that means we can here directly show that the physical properties of the cells have a direct consequence on how the cells circulate and which organs they reach and where they eventually get stuck. So modulating mechanical properties that we can measure in RTDC might be an interesting way to address circulatory problems. I think I still have a few minutes, so I want to quickly switch gears also to show you some technological advances. I mentioned FACS earlier. Of course, we can also introduce a fluorescence measurement here, 3-color fluorescence, and interrogate the cell for their fluorescence signal. At the same time, we set this up in a way that the cells are moving through a light sheet. And as they pass through, of course, there's a temporal signal that translates directly to the distribution of fluorescence within the cell. So if you want, this is one-dimensional fluorescence imaging. And then you can, for example, see that in the different stages of mitosis, the metaphase, you have the chromosomes in the center, and then you get one peak of fluorescence in the center, or at the end of anaphase, the chromosomes have separated, and then you get 2 fluorescence peaks. So this just shows that you can do one-dimensional fluorescence imaging also in real time. And now we have a situation where we can do normal FACS analysis, but also this image-based analysis for mechanical phenotype in this channel and in real time. And that means we can use the information that we gather in real time to actuate a trigger that will then sort the cells into a channel. Cells of interest can be sorted out using surface acoustic waves. This works for fluorescence. So I plotted here as the area of the object, the size, versus fluorescence intensity, do 2 different beats in this case, low and high fluorescence, and we can take out the cells that have a fluorescence expression with very high accuracy. So that works well. But now we can also do that for deformation. So you mix 2 kinds of beads with different stiffness, and we can take out, for example, the stiff one or the soft one in a separate experiment. So that's really cool, because what that allows us to do now is to do things like you have this distribution in mechanical properties in a population, and you can then choose to take out the cells that are soft or the cells that are stiff. And once you have them separated, you can then analyze them for what makes them different. Right? You can do a RNA-seq analysis to identify the transcriptomic profile of soft and stiff cells and compare in order to identify maybe novel ways to regulate the stiffness of cells. Right? That's kind of the goal of that exercise. This is one of them. We have a couple of others in the endeavor to getting control of cell mechanical properties. For number 1, showing that they matter, right? If you could arbitrarily change cell mechanical properties, you can also test in, I don't know, in a developmental system or in other migratory context that mechanical properties are important, or even in the medical arena where, maybe cells are too stiff and then get stuck in the lung and cause problems. If you can specifically tune the stiffness, make them softer, you could also alleviate some of these health aspects. So I think this is really an interesting area that we're moving into, where this is not just a phenomenological property that we can measure, but it's a property that we start to get control over. And in the remaining 2-3 minutes, I want to address yet another interesting point. I think this is also interesting in the context of the PTB, because we have in over the course of the years seen that there's a problem when you do measurements of cells, in that you take one single cell type, split them, and measure them with two different techniques where you really know everything about the stress and the strain and everything, and in the end you get different results. This is something that we published a few years ago with all kinds of colleagues, with all kinds of techniques across the world, with a single cell line out of a single stock. And the results were super surprising. If you're interested in that, I highly recommend that you read this paper. And basically the upshot was, amongst other things, that the measurement time scales are really important when you measure viscoelastic materials such as cells. So if you have an AFM and you push down on a cell and you think that's an elastic object, you use a Hertz model, you extract a Young's modulus. It matters whether they do this fast or slow, and then you have a different technique like an optical stretcher or something else, and then you will get very different results simply because you have different measurement time scales. That's also the result of the other study with the microfluidics that I mentioned earlier. In order to circumvent this, to have something that you can use to compare different techniques, we came up with a standard, a cell mechanics standard, that we produce from polyacrylamide by a microfluidic droplet generator. So you push in the pre-gel mixture into a junction with oil to form emulsions, droplet. And because of this microfluidic droplet generator, the particles are all very, very monodisperse. So you can tune the size of these particles by the pressure that you apply. And you can go from, say, 10 to 20 microns roughly. And obviously, the higher the pressure, the faster the production frequency. So you can make a few 10,000 beats per second and doing this way. We also characterize them with various techniques that we have available, such as optical diffraction tomography and Brion microscopy, to show that they are really not just spherical, but they're also homogeneous on the inside from their density and also their mechanical properties. So these are really homogeneous spheres, monodisperse. And then we also compare different techniques to measure their properties, in this case using atomic force microscopy, but also these microfluidic techniques with different viscosities and flow rates. So with ultimately the different shear rates to show that they are essentially giving us the same Young's modulus, which means that these are really elastic objects that are independent of shear rate. And with that, we finally have a calibration standard, not only to compare different techniques against each other to see whether they actually agree on certain types of measurements, but also something that we can introduce into a system such as the RTDC before every measurement to just make sure that it operates to a certain standard, and then can compare the results that we're getting with cells against that. This is something that is available as a resource to the community if anyone is interested in playing with these speeds. So with that, I'm at the end of my presentation. I think the take-home messages could be that this approach of physically phenotyping cells is a sensitive and marker-free way to characterize cells, and in particular to also detect functional changes in cells, physiological changes, but also pathological changes. That this RTDC technology is a relatively new high throughput way to do that, to exploit that, and even move that into the area of cell-based diagnostics. We can combine this physical analysis with fluorescence detection for additional molecular specificity and also sorting, which opens up very interesting new ways of thinking about cell mechanics. And the last few slides I also showed that we have now some calibration standards that are available to mimic the mechanical properties of cells as mechanical objects. That's my group, collaborators, funding, some companies that we're having fun with. And if you're on Twitter, we have a Twitter handle so you can follow what we're doing. Thank you very much.
Yeah, thanks for this really exciting and interesting talk. I'm really impressed how you put together photonics and microfluidics, and microfluidics is even not very complicated, to get really new insights into mechanical properties of cells, and to connect this to the phenotype or the biological properties of cells. Yeah? So I wonder. This is probably a long learning curve needed to connect really these mechanical properties to the biological features of cells or disease. This is not very common so far. So, so hopefully this will be not too long. And in the beginning, you started with different definitions of cells. Are these really? I think this, if to my understanding, this is complementary information, isn't it?
Yes, I would think so as well. I mean, definitely it makes a lot of sense to define cells by presence and absence of certain types or groups of molecules. Definitely, right? I don't know. An immune cell will have certain receptors that are required for an immune synapse that will be characteristic for that cell that, I don't know, a neuron will not have. Sure. But there's additional information that you can get, additional information that will help you to better characterize the cells, to not, maybe there is a cell that has the same kind of surface receptor, but it's physically very different. So you can then distinguish them, right? That's something that you get in addition and for free by using that kind of approach.
Okay. So there are some questions, I guess. So I start from the top with a hand waving. I didn't recognize who was first, so maybe I start here, PTB Abteilung 8, or who's that? I think Tobias.
Yeah, that's me. Sorry. Yeah, thank you very much indeed. Again, this is really interesting. It's fantastic movies, I have to say. It's really cool to see how the deformation is also taking place in the kinetics process. Fantastic. Huh?
Yeah.
And I see already a number of connection points to the stuff we are doing here in cytometry. So a little bit question which has been already raised, and maybe following up on that, is the differentiation. And my feeling at the moment, if you're talking to lab medicine people, the most of the things is done by fluorescence. If you're thinking about the leukocytes, then we have the CD3, and this is always the antibody binding things which we are having, and or CD4, and there are a lot of bits there. And there are other people they're using impedance cytometry, having a different marker on that one. So what is the differentiation if you with the mechanics in comparison to other physical properties like absorption, scattering, impedance, and fluorescence? Is it really making a big thing, or is it something which is doing a little bit? And I can understand your point in the area of label 3. That would be fantastic. But as soon as you go in fluorescence, do you have not already everything together what you need?
Well, I mean, that's the point I wanted to make right at the beginning with the tomato. Two tomatoes might be red and round and everything that you can measure in physical measurements, but you want to know the mechanical properties also. And there will be difference. And so you will keep you from buying rotten tomatoes. That's the difference. In very blunt terms, right? Of course, I was trying to point that out also in the context of the circulation, right? This is the most obvious way where size and mechanics will impact how the cell will get around your body. That is not something that you can get from a fluorescence readout or an impedance readout, because that is not connected to this particular function of the cell to be able to move through the body, to be able to experience forces, stresses that need to lead to mechanical deformations, right? Shape changes. And shape changes are important in what the cells are doing. If the cell cannot deform its shape, it will not fit through a small hole. It will not be able to extravasate as quickly, as to migrate as fast. I think there's a very direct connection between the mechanical properties of cells and what they're able to do in the body. Because cells are not just places where biochemistry happens, right? This is not just molecular biology. These are also physical objects that where the physics impacts what the cells can do. And that's the technique to measure that.
I understand. Thank you very much for again clarifying. So what's your experience with the medicine people, are they asking for that? Is it something which they're requesting, or is it something?
This is not something that they know, obviously, right? They're not exposed to that anywhere. But as soon as you tell them that this is possible, they immediately click. I mean, this is something that you don't need to sell them, because you know, I don't know, you can feel a tumor in the breast, palpation. So the haptic sense is not something that a good doctor would be not familiar with. The good doctor uses that kind of information for diagnosis.
Yeah.
Tell them, you know, you talk to surgeons, they say you can feel the margin of a tumor. You push against it, you feel there's a mechanical difference. And this is a way to quantify that, right? This is a way to bring that down to the cellular level and get that kind of information quantitatively and in high throughput about cells. They immediately understand that.
Okay, so I would like to give chance also to other colleagues for questions. So there's one colleague, Hugalf.
Hello, can you hear me?
Yeah, more or less.
Okay, nice. Thank you very much for the presentation. It was very nice and cool and a nice insight to your work. My question is whether you have ever tried to make a real-time sorting, kind of you take blood from a patient and then sorting out, like for example, metastasis, or like bad blood and give it directly back to him?
Yeah, that's a good question. So no, short answer is no, we have not done that. The throughput is in terms of cells per second is impressive, but in terms of volume processed, it's really tiny. Right? If I said in the beginning, we take a few tens of microliters of blood, and then we dilute that, and then we measure that for hours if you want to, but typically 10 minutes or 15 minutes. And then we've processed, I don't know, a microliter of blood.
Okay.
That would be not that the throughput is just not there to do anything in the volumes and the quantities required to really, you know, do, I don't know, apheresis or something, anything, something like that, right? To just rinse the cells, the blood of bad cells. And then the problem with circulating tumor cells, because you mentioned that, is even more dramatic because there are only a handful of circulating tumor cells per milliliter. So these are really, really rare events that we can detect if they're there, but we probably never see one. So that would not help in a blood cleaning setting.
All right. So next question may be asked from Mr. Zack.
Thank you very much. Sorry, my internet was left for 10 seconds. But
Jochen,
Ingo,
very
shoot.
cool talk. So can you hear me? Yeah, I think so.
Yes.
So I like the technique, which I don't need to mention actually. So I'm a fan of, which you know, we already use that to study and understand the effect of gadolinium-based contrast agents to leukocytes. So here we found the major effect in leukocytes and not in red blood cells, other than in the COVID-19 case you have shown. The red blood cells constitute the majority of the blood. So, and I'm asking myself, would you expect to see on the macroscopic properties of the blood already a change there? Could you compare this simply with viscosity of the blood, finding that the blood becomes thicker, more clotted, which is an obvious finding actually with COVID-19. So wouldn't this not be much simpler to detect COVID-19 with blood viscosity?
Yep, might be. I've also heard that anecdotal reports that the viscosity of blood in COVID-19 and also long COVID changes. And in particular, there's also microclotting going on. It's a big topic in this context as well, right? There are thrombotic events that also lead to further problems, etc. I think there might be simpler ways to measure viscosity of blood for sure. I'm wondering whether that is the only thing that you want to get from that situation, because if you get the changes in the red blood cell properties which translate into changes in viscosity, but you also get the information about the white blood cells in addition. I think there's just more information that you can then use to interpret this particular situation at hand and to be maybe more specific in the diagnosis. In particular, you can probably detect a small fraction, 5% of abnormal red blood cells would be obvious to detect with this technique, which might not yet have a strong impact on the viscosity of the cell, of the blood overall, right? So it might be easier it's a more sensitive way to get at the core of what's going on. And also it will give you a direct access to figuring out what the mechanisms then are, right? Why are the red blood cells stiffer or less deformable? And why are particular other cell types activated or not activated in particular ways, right? So you get more closer to the actual mechanism of the disease. So I'm not saying it's a stupid idea. I think it's a great idea actually if you can really diagnose COVID-19 or long COVID by blood viscosity measurement. I just think that is relatively blunt compared to what we're doing.
Yeah, I agree. So I heard it's a general pattern of virus infection to clot the blood or to affect blood viscosity here.
Yeah.
I believe that you are much more specific here. Thank you again.
Thanks.
Yes, so I would like also to put in some questions from the chat, very early questions. So Claudia Swart from PTB asked, also connected to the COVID patient samples, if your method can be used to predict the severity of the disease or the cause of the disease. Is there already some indication for that?
We have no indication, but I think that might be possible.
Okay.
So in that study, we looked at 17 COVID-19 patients, and they were at different stages of the disease. Some of them died, some didn't. They also had different ages. They were typically above 65. So this was relatively early on when mostly very old people were succumbing to that disease. So we didn't at the time think about that question already. So otherwise we could have tried to maybe also look at severity, but I think you would need to do a much larger study to get at that. But I think it could be used, yes.
Mm-hmm. Okay, and there was another question, does the orientation from Jakob Jordan, does the orientation of non-spherical cells going into the channel influence the measurement in any way?
Yeah, definitely, right? So most cells, if they are in suspension long enough, they will become spherical. Then that doesn't apply, but there are some cell types, in particular, it's also activated in blood cells, leukocytes. They can have, I don't know, pear shapes and things like that. There could be some pseudopods and protrusions. So they're non-spherical. And then, of course, they will orient with their major shape axis along the flow direction. So they will kind of flip around and move through in this orientation. This will, on the one hand, this helps, of course. So if you have all the objects oriented in a particular way, it simplifies the measurement and the characterization. It will prevent us from calculating a Young's modulus because we can only calculate the stresses for spherical objects, initially spherical objects that move away from sphere in small ways. But we can still characterize them in any way that you want, right? You can say, oh, look, the aspect ratio is larger or smaller between different samples or patients or treatments or whatever. You can look at other features, texture features of cells, in cells. You can even use the images directly with AI to compare, classify cells and compare them in different situations. So cells being non-spherical only prevents us from calculating a Young's modulus, an apparent Young's modulus, but other than that, we can still characterize the cells and their deformation.
Okay. Okay, there's more hands waving. Christian Görke has a question.
So thank you for your talk. I was wondering if you destroy a certain amount of cells using real-time deformation cytometry. So do you create some debris in the system?
No, the measurement itself does not create debris. There might be debris in the sample because of the preparation, pipetting, whatever, or there are dead cells that are already disintegrating, but the deformation itself is gentle enough that it does not destroy cells.
Okay.
Obviously, if you have a very soft cell and you have very high viscosity in the medium, you generate very strong shear forces, red blood cells, you could probably tear apart. But then you have two parts and not really debris in that sense. And that's kind of moves through and then it's gone and not analyzed anymore.
So do you have to adapt your system according to your sample each time?
No, not very much. We have a typical size diameter of 20 by 20. We also have 15 by 15 and 30 by 30 and 40 by 40 if we really want to measure very large cells or much smaller cells. But typically it is 20 by 20 and there's one standard buffer that we use and one standard flow rate. That gives you typically enough bandwidth to characterize most cells, even in blood. I mean, platelets are too small. They are not much deformed. So that's hard to measure mechanical properties. But then from lymphocytes to the myeloid cells to the red blood cells, you can nicely characterize all of them despite their stiffness and size differences in a single go.
Okay, thank you.
There's one more question from Jonas Ginger.
Yes, thank you. My question, or actually two questions, but the first one is just a quick one. What do we have to do to get our hands on these calibration materials that you mentioned?
Send me an email.
Okay. And the other is, you said that timescales for the deformation are important when you compare different measurements or when you want to measure certain quantities. What would happen if we somehow try to measure deformation in a more regular flow cytometer setting with higher flow rates and shorter interaction times? Would that make any sense, or is it, if the interaction time is, say, I don't know, factor 10 lower, or is it pointless to actually think about it?
This is not pointless. So you're thinking about a flow cytometer where you also reduce the channel size to a size where there's hydrodynamic stress acting.
Yes, I mean, we have a different kind of hydrodynamic stress in a regular cytometer where we don't have the microfluidic channel, but just assuming that we can somehow generate some sort of stress somewhere, maybe not in the channel, maybe before the channel or somehow, could we get something reasonable if we have just higher flow rate and a shorter interaction time, or not?
Flow rate and interaction time are two parameters. You want to also consider the viscosity in that case, because what ultimately matters is the shear rate, and that depends on the viscosity as well. If you go up by a factor of more than 10 to what we're doing, you will run into a situation where you can depolymerize actin, for example, which means that you're not sensitive to some things that have to do with actin cytoskeleton, like cancerous changes. But I mean, there's also a question related to that. Obviously, there are other structures in the cell that also contribute to the resistance to deformation, such as the presence of a nucleus. Once you start deforming cells more, you will probably also run into resistance of the nucleus being deformed. Right, so you become sensitive to other things that might not be easily accessible with what we're doing. You can play with viscosity, so you can speed up the measurement, speed up the flow rate, and reduce the interaction time, and still have the same shear rate by also appropriately scaling the viscosity. And we elevate the viscosity to about 15 millipascal second, so that we get a reasonable shear rate, also shear stress, but don't have to move the cells so fast they become blurry, because we still want to take images, right?
Okay.
So I'm not sure if that answers your question, but number one, I recommend that you read the paper that I pointed out, Obanska last year. And if you have more questions, I'm happy to discuss that offline.
Thank you.
Okay, thanks. So there's one final question from the chat. I would transfer this. Is there techniques that can differentiate between cell body stiffness and nuclear stiffness?
Yeah.
To understand the contribution and the overall stiffness.
This is a really good question. So as I just said, the cytoskeleton is one major component that will control or contribute to the resistance of cells to deformation. In particular, when you start deforming cells to beyond a certain amount, you will also run into the problem that there's a nucleus that needs to be deformed that will contribute. Example, the malignant transformation is a very good and relevant example here. The problem is that we don't have any simple means of distinguishing the contribution of a nucleus or a cytoskeleton because we're blind to that, right? We have only the overall shape in the way that we're doing this at the moment. And without any additional information, you would not be able to distinguish the difference, where the contribution comes from. You can do things like, I don't know, compare a sample with and without actin depolymerized with cyto D or latunculin or something like that to take away some of the contribution of the actin. That's one way of doing it. Something else that one could do, I guess, is also use the fluorescence capabilities of the setup, because I said there's a one-dimensional fluorescence imaging, so you could compare the signal of a cytoplasmic stain to the signal of DAPI or Höchst, and then you at least know the relative size, and then you can probably come up with models that take into account what a larger nucleus with certain properties might do versus the cytoplasm around that. So that could be possible. This is a good question, and I think this might be an interesting area to explore, but we're not doing that yet.
Okay. So I think that was a very fruitful and interesting discussion. So thanks to our speaker again, and thanks to all the audience to our seminar. And I think we will close now. It's time is enough for this colloquium. So thanks to the audience, and I would appreciate if Jochen Guck and Tobias Schäfter, if we can have a brief chat as a follow-up immediately. And I thank for all the other participants and wish to have a nice day. Okay.
Thanks for joining. Thanks for your interest.